I titled this as image processing, this session as image processing. We look at how image processing is done within the human system, and how it would be similar to image processing in engineering, and what are the possible similarities and differences which exist. I will try to make a highlight of it. And in fact, once upon a time when the biological part of image processing was taught, you had to accept things at face value. In fact, you would realize that. But having gone through image processing techniques, I understand that in fact you find neural network techniques as a excellent methodology which says that this is the same thing which is going on within the human visual image processing architecture. So, it is a two-way communication. Unfortunately, the two-way part of it has not happened. Most of it is the engineering side taking ideas from human vision and monkey vision and trying to implement, and with spectacular results which are aware. So, I put it as image processing. We start with some basic architecture. You are familiar with these things in the eye, retina which has got the circuitry which have got multiple cells. The cells are actually in the way of light, and the receptors are the depth and the receptors as opposed to conventional neurons are neurons hyperpolarize as when light photons impinge on them, and that in turn causes reduction in receptor activity. So, when receptor activity is decreased, the output cell, which is the ganglion cell, the output activity increases. So, that is how signal is generated in the on stimulation with light within the visual pathway. So, with that introduction, I think I will start with network analysis. Network again taken from Candle and Schwartz. Please go through the textbook. I am walking through that because the network is, I think there is an elegant depiction of the network, and this network is important for subsequent discussion. So, cone signal and rod signal circuitry. We will start with the cone data. So, two adjacent cones. There is light only on one specific cone over here. What happens with that? Now, things in red indicate that there is negative, the other one is positive. I think I have only one color, so it is red again, and it would be confusing, but I think the pointers are very elegantly placed enough to convey the data. Now, one of the things which I had discussed, one key point which needs to be remembered is there is no 1:1 data transmission. So, one cone does not go into one cone goes to one ganglion cell in the fovea, but it is not necessarily a feature. So, how the architecture and it is not a linear transmission. So, you have got several cells in between, multisynaptic pathway, multisynapses indicating multiple other cells. We now discuss the roles of these cells. So, a single cone, and the single cone which is over here stimulates a bipolar cell, bipolar because this cell has got one process over here and another process over here. This bipolar cell in turn reduces the signal over here, and that in turn causes ganglion cell. Ganglion cell is the output cell of this entire architecture, it is an on. Now, it also implies that this cone sets up another pathway of the adjacent cone in which a bipolar cell is inhibited. This bipolar cell in turn causes negative feedback on amacrine cells. The bipolar cell also inhibits the ganglion cell, but because the amacrine cells are also involved, there is an off. So, one photon causes one ganglion cell to be on and the adjacent ganglion cell to be off. So, not only that, This cone also through these horizontal cells signals these horizontal cells which are involved in subsequent pathway of the adjacent cone negative, by which it causes something called lateral inhibition. So, what happens is you have a central on and then there is a next to adjacent off. If you look at rod signal circuitry, it is somewhat similar. There is cone stimulation, which in turn causes this bipolar cell to be activated and ganglion cell activation, but the adjacent ganglion cell is off. Rod signal bypasses through the rod bipolar amacrine cell and then causes two signals, one negative over here and a positive over here, positive in the sense of ganglion cell activation. Now, what this means is better explained over here. So, this is the same concept in a different picture, and this is what is I think better represented. So, again if you look, the idea that light has to traverse through the entire cell structure is shown over here, goes and stimulates the a set of photon. It is not a one photon to one receptor. So, there are many photons which stimulate a set of photoreceptors, and all the photoreceptors are some of the photoreceptors are stimulated, meaning it based on intensity of the stimulation. So, there is a set of cones which are cones, or in this case it is rods which are activated, but this implies that because of the secondary circuit over here. So, this secondary circuit ensures that there is one set of ganglion cells in the center which is activated because of this, but it also ensures that all the peripheral in the sense next to this adjacent is a better term, all the adjacent receptors are inhibited. So, you got a central activation and a peripheral inhibition, and remember this is at the level of one single set of photoreceptors. We have 6 million or something photoreceptors, and this is what is the status. So, if we look at signal generation, so signal generation would look like this. So, there is an on center and an off area by which diffuse, say for example, the native signal may be something like this. The native signal is something like this, but then it has got focused into this specific thing. It does not activate this entire thing. So, it is this signal is converted into a very specific signal by which you have started the beginning of edge recognition and contrast. So, these are for sets of receptors, please remember. So, these are 3 dimensional, and edge recognition is basically you have formed an edge between a light area which is lighted and whereas an area which is not lighted, and that is the principle by which you have the on area in the center and the off area in the periphery. So, that processing in turn is mediated through multiple channels. One is of course mediated through the same pathway, but one is the the ganglion cells in the next level on the sides are inhibited and the central cells are excited. So, that is the idea, and the other method is that these cells which are in the periphery are inhibited, whereas the center is stimulated. So, that is a important concept which is there, which is present within the first level of this is retinal signal processing. So, retinal signal processing is very sophisticated. It is the constructs of image processing such as edge recognition, segmentation, multi-object segmentation is already started within the retina, which is not just a dumb sensor. A sophistication which is far more than any of the devices which I am aware of. So, this is something. Now, I spoke the idea about the idea of multiple receptors converging onto a single ganglion cell. So, the fidelity of transmission obviously depends upon the number of receptors converging into one ganglion cell. So, that is the concept which is highlighted over here. In the periphery, you have very large receptive fields. So, what that means is that anywhere in that area you put in a stimulus, you would get one ganglion cell activator. It is just a numerical way of putting it. So, you can say that there are about 100 cells over here, and that leads to one ganglion cell which is activated. Whereas, when we look at the fovea, you have very small, see here actually it is a hexagon. So, one positive which is surrounded by 6 negatives, one positive 6 negative cell, and that goes to one ganglion cell. So, there is a lot of fidelity differences between the peripheral retina and the central retina, which is the fovea, and that is what is depicted over here. So, now the area which is supplied by from which data is collected by a single ganglion cell is the receptive field. Receptive fields of the receptive field of the ganglion cell, and that is how the architecture is built, or rather it is present within the retina. So, now it gives rise to a lot of downstream discussion, which I think I have taken these pictures from various sources, common media sources. So, these are the pictures. So, suppose you have a central area of lighting. So, the stimulus is like this, and you generate responses which are there during the process of lighting. On the off area, it is for this is a specific receptor field. So, you shine light in the center, this is the response which you get. So, light is in the center, and for a light which is in the center, there is a peripheral off. Now, if you put light in the peripheral off, you have a signal which is out of phase when compared to this signal, which is outside of the receptive field, no signal, entire area stimulated, no signal. So, no signal when signal is either outside of the field or completely this one. If we put part of the field, again it would look somewhat like case 1. We skip to the next one. So, I think the right title for this paper should be Boolean algebra in the eye. I would invite enthusiastic listeners to model this somewhere. So, let us see what happens with an on center and off center, light on in the center only. So, light is on in the center, ganglion cell fires rapidly, ganglion cell is the output cell. So, this is on center cell. So, there is an off center cell which fires when the center is off. So, this is on center. So, on center cell is with reference to ganglion cells. So, ganglion cell on and on center, off center. So, on center means light is in the center, off center is light is in the periphery. that sort of reflects the idea which is over here. So, we come back to this discussion on center cell, light on in the center only, ganglion cell fires rapidly. Off center cell, ganglion cell does not fire because light is only in the center of the receptive field. Light on surround only. So, the off center cell, off center ganglion cell fires rapidly. This cell does not fire, which was on center. No light, obviously nothing fires, not fire, not fire. Then light on center plus surround, center plus surround. This is center plus surround is weak response, weak response, weak response. So, should I attempt center and off center? Case 1. Is it, yeah, we need to put something. So, is it a function? It is not a function. So, light center, light in the center, no light in periphery implies on center plus ganglion cell on center plus ganglion cell off center negative. Case 2, light negative of light in the center, but light in periphery implies ganglion cell on center is negative and ganglion cell off center is positive. Case 3, light in center is not there, light in periphery is not there, implies I think I should work on the nomenclature a bit more. Negative comma G of negative. Case 4, plus light in the center and plus light in the periphery implies ganglion on as plus star comma ganglion off as plus minus. So that's sort of, you know, you've actually done image processing and a lot of gating done with this diagram. That's why I thought I should include this. So so what happens is larger context. So this is the stuff which is I think it's there in the textbook, that is the point. So there might be many other kinds of processing which I am not read about and which I am not aware, and I can't be telling you things which I am not aware of. There would be other kinds of processing which are there, but the highlight of the entire story is that there is a lot of you know okay, I think I should state this. An image is actually not formed in the receptor level. The receptors just take in data, the data is immediately processed. So, there is never an image within the eye, you know, at least, you know, if you look at movies in which you take data from the eye and reconstruct images, this just goes against biological logic, that goes against biological logic. Biological logic is it is just pure data. The retina takes data and then not the retina, the receptors take data, passes it on to the cells. The ganglion cell, which is the first level, already gets processed data, it is not raw data. It is not just the photon intensity, it is not the color channel, not just the color channel intensity. It is a lot of other, it's already changed, and that there are different cells which are hardwired for these purposes, and combinations and permutations of this. So, there is already a neural network in the retina, so as to say, and the first level parsing of the image happens over there. the image as such is not formed. So, if you can't say that this is the image in the retina, this is the image in the optic nerve, this is the image. So, there never has been an image, that is a key thing. So, this is something which I think I will skip. So, yeah, so this is the next part of the story which I have to tell. So, what happens so far I have discussed about what happens within the eye, then I have described stuff which happens in the chiasm. Now, I included this slide in the image processing part of the story for a specific reason. Now, the LGB for instance has as a very funny thing which I do not know how to interpret this. This is one of the places where I told that engineering methodology can show or shed light into biological processes. See, so far in the introduction I told that it is engineering which has been inspired by biology, but there are places where biology may or should be taking in data from the engineering side and work out methodologies by using engineering methods, techniques, algorithms to interpret what is found in biology. Now, the lateral geniculate nucleus is designed in layers. So, we have 1, 2, 3, 4, 5 and 6. Now, optic tract is the input, and then there is this layering which is happening. Now, note that I have told that half of the data is coming through from the optic tract from the opposite side, half of the data is coming from the same side. And what is shown over here in two colors is this: contralateral means opposite, ipsilateral means same side. Now, 1, 4 and 6 layers 1, 4 and 6 take in contralateral data, 2, 3 and 5 take ipsilateral data. So, this would mean opposite nasal field, same temporal. If you look at it in eye terms, when I told you about field, remember that this is coming from this entire LGB of one side is taking data from this side of the field. So, that is what is drawn over here. So, this is the opposite field which is conveyed to this side lateral geniculate nucleus. Now, this particular funny nomenclature, 1, 4 and 6 is odd, meaning it is definitely numbers are even, but it is odd. 2, 3 and 2 plus 3 is 5, that is how you remember for writing entrance exams. So, 2, 3 and 5 is same side, and the importance of this is it is not just a biological curiosity. The same representation carries on all the way up to the cortex. So, that is the significance. So, this is the visual cortex, and this kind of representation, it is coded. Okay, to put it to explain it in another fashion, parts of your field are coded all the way from what part of your field is being seen all the way up to the cortex. That location is mapped, and maybe that is a and it is with high fidelity, that is the point. So, LGB is fairly high up the hierarchy. So, this data from a particular point in the visual field of a person is taken, and there are specific sets of nucleus within the LGB somewhere over here, and then it goes all the way to specific layers within the visual cortex. So, there is mapping, and to recollect, to re-jog some of your thoughts, if you remember, the same idea is there in the spinal cord, in which fibers from the leg which go first into the center of the spinal cord, and then the hand fibers go into it, and then that gets carried on upwards. So, this idea of locking in spatial data of varying kinds, in terms of sense organs, it is data from the foot versus data from the hand, which is which is anatomically preserved and taken up. In terms of the eye, it is data from one field which goes to the opposite side. Even within a particular field, that is left side field or right side field, one of the fields, there are areas which go to specific layers of the LGB, and from there go on to the cortex. And if you see some of these CNN designs, they have been inspired based upon this. But CNN designs are very straightforward, they have very simple notions of this complex architecture, and I do not think anybody thought of implementing the 146.235 architecture for that. There is a relevance, I am not able to find what the relevance is. Now, if we look at architecture further on. So, if you look at visual cortex as such, so visual cortex as such contain the whole thing is a cortical column, cortex rather, and within the cortex you see these bands over here. And I think you should also notice that this is a very old paper, 1988, and I used it for that reason. So, the people who are actually in the field should look back into these things, and you would get novel ways of looking at image processing with these things. So, I will just read it out. A cortical computational module, a chunk of cortical tissue 1 mm in diameter, contains an orientation hypercolumn, full cycle of orientation column, one cycle of left and right ocular dominance columns, blobs and interblobs. This module will presumably contain all of the functional and anatomical cell types of the visual cortex and would be repeated hundreds of times to cover the entire visual field. So, there are cells which are for orientation, then there are cells which are for dominance, left eye, right eye, and then there are these blobs. So, all these various kinds of data, I think I missed something earlier. Yeah, so this is the slide which I missed. So, again people who have looked at large what is that deep neural networks with hundreds and hundreds of layers are familiar with this concept, meaning they would have read it that somewhere down their ANNs they start to form neurons which purposely look at orientations of cells. Now, that actually came up, as I understand neural networks, that came up as a function of back propagation. It was not designed to be implemented like this. When they have done back propagation and then taken intermediate layers for analysis, they found out that specific layers, specific neurons, artificial neurons have this property that single neuron would get excited with a particular orientation of a line, a curve, a color, or something like that. That's how CNN is built. Now, if you look at human cells, they have the similar property. So, this is I think cortical cells, and when there is a particular orientation of cells, there is a when there is a particular orientation, there is inhibition and then there is activation in all the other orientation. So, that would sets of neurons are there which get higher level. This is not at the retina, this is at cortical levels. There are sets of cells which can be shown to be happy when there are lines of a particular orientation. So, that is one of the things. So, now we look at different paper. This is somebody from 2000, and how orientation preference causes change in spikes. So, let us look at that. Orientation which is like this causes a signal which is happening over here, but when there is the other kinds of signals you do not have. But what happens is when you have moving cycle of movement across the field. So, you have the neurons perceiving the movement and the direction of the line. So, that is I think I will again read this thing. Vector averaging as one model of a population coding in neural circuits. Vector averages describe possible relationships between blah 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 and sorry, meaning it's the stimulus orientation to which a neuron fires can be thought of as a line label. When the cell fires briskly, it activates activity signifies the presence of a stimulus with that orientation. So, orientation can be coded on to individual cell firing, and that is something very interesting, which is there is a 2000 paper way before AlexNet and CNNs. So, way before in 2000 experimental evidence is there that this kind of pattern happens, and now we are seeing that CNNs actually generate neurons with same interest. So, there is some fundamental thing. So, though back propagation does not exist in conventional neuronal biology, the neurons do not solve differential equations back and get you weights and what is that weights and biases adjusted. There are mechanisms, maybe that this the network had done its homework a couple of million or billion years back, but we are seeing the final robust network, which is the network which has been found to be useful, and these networks, these biological networks show phenomena which have been demonstrated in CNNs, and that I think is a very important reason why I just put the slide. I just put the diagrams and I leave it for the audience who are interested to go in greater detail. So, another kind of data, I think this is again from Candle. So, free to look up the original paper. So, we are looking at cortical layer data, and what actually happens with these how movement is perceived, I think. So, you have stimulus which is going from here and then turning signals which have actually which are not there here and which are not there here, but you have got robust signals over here. So, which indicates from the receptive field data that there is a line in the which is the interpretation of a line. So, receptive field on, off center, all that stuff gets this is retinal level data. So, retinal level data to higher cortical association and the in between stuff. So, picture which shows all of that data and how movement, how orientations are perceived by using neural networks, and how receptive fields in the retina which are generated have been transmitted all the way up to the cortex, and the cortex interprets this as a moving line. So, this would form I do not know what is next after CNN. There is so many of them that I do not recollect the name offhand, but I am pretty sure people who are in the field would recognize something very what you call it looks like a CNN architecture. Then what is this? This is monkey, I do not know, some animal model where there is retina, lateral geniculate nucleus, the primary visual cortex and secondary visual cortex and the temporal pathway. So, different kinds of data, color, depth, direction, orientation, complex forms are generated at various levels, and say for example, color here, then there is direction here, goes to a different area for direction mapping, color is processed somewhere else, and what is this, complex forms are processed somewhere else. So, this is how the visual system perceives objects. I think I should reiterate here with a greater confidence that images as such are not formed anywhere within the brain. So, it is something else. So, you do not take like a camera, you do not take an image and store it in the brain. Its attributes which are stored. So, these attributes are blended with several other kinds of attributes from several other kinds of sensations and mixed up. So, that is the other slide. In between, I think I should, because the topic is the same. This is stereopsis and binocular disparity. So, how is depth and so many other things, sizes, depth, everything perceived. So, binocular disparity is the difference which you have because you are viewing from both the eyes, and then there is the difference in focal length which is interpreted. So, how that is interpreted is what is shown 1995. So, very interesting that many of these things I do not think even now maybe these algorithms have been put into practice in biological in engineering context. I have not read about anything in the CNN side which is looking at these things. So, depth is computed from the positions in which images occur in two eyes. So, two eyes is the key word here. The image of the object lying in the plane. So, you have a plane which is there, which is the focal length for the retina at that given point of time. Fixation green falls on the corresponding points of two retinas. The images of objects lying in front of the plane of fixation blue or behind it yellow fall on non corresponding locations. So, they do not fall on the same area, they fall away from the primary fovea, which is where the density. So, that is used to, so that causes binocular disparity. So, binocular disparity is between the difference between this and this. Visual neurons are selective for particular ranges of disparity. So, that is a key term here, range of disparity. Each plot shows response of a neuron to binocular stimuli based on the deviation from the center. Some neurons are tuned towards a narrow range of disparities and thus have particular disparity preferences, tuned excitatory or tuned inhibitory neurons, whether others are tuned broadly for objects in front of the fixation plane or beyond the plane. So, you have mechanisms or not, they are not mechanisms, they are neurons which are happy when there is something in front and there is something behind your field of focus, and they are happy about it. They are not bothered about whether the object is of interest to the person, not of interest to the person, they are just excited. But when there is something else happen and the focus needs to shift from the immediate focus to the next point which is there, this neuron would help you to tell that the next level of focusing has to be towards you or away from you, whether the eye moves away to get focus on a distant object or whether eye has to move towards each other to fix on a closer object. So, that is the idea. So, these are maps which are there within the cortex. So, I will just list out. In fact, I myself have not read this paper, so I do not think I am an expert on this topic, neither am I expected to be expert on this topic, but my idea of conveying it to you is that these things are there, and for people who are interested, please do look up, read, because they are interesting to read if you have the time and necessity of reading it. So, what is shown here is ocular dominance columns, orientation columns, which is all color coded. So, various parts of the cortex are hardwired to different orientations, and then there you have these architectures of blobs, stripes. These are higher level computing architectures, maybe something like equivalent to server architectures in computation. So, you have different sets of servers computing different things and they are all wired together. So, something similar to that, you have blobs which contains aggregates of neurons doing some specific function, and they do it without they are not they do a specific part of the task, and that is the similarity between CNN deep layers, where specific layers or specific neurons within the CNNs take in data for a specific task and then just pass it on to the next layer, and you get a defined output at the end what you want. But that again remember is back propagation, and this is not back propagation. So, this part of the discussion would be incomplete without discussing higher level visual processing. So, we have low level visual processing. So, image contrast, see on off center, on off, and on center, off center gives you lines, boundaries, contrast at the level of the retina. You also have data on color because ranges of receptor function correspond to color. So, you have a particular say you have I think I should draw this, that is why. So, you have a photon of this intensity, this intensity on that. You have two cones which are side by side, and one cone will stimulate at plus 3, and then you get plus 2 from the other one, and these two go to ganglion cells, one and ganglion cell 2, and then you have a specific color coding. So, I had not mentioned this earlier, so that is the reason I told. So, a photon of this particular nanometer impinges onto the retina. So, that would stimulate one cone which has a spectrum like this, and another cone which has a spectrum like this. So, this is just looking at color, it is not looking at spatial orientation, which I have discussed so far. So, color can be coded like that by differential actuation, differential stimulation of two sets of receptor, two colors receptor. So, this is green and red. So, green and red stimulators receptors are stimulated to varying extent for a given single set of photons, and that gives a sense of what nanometer of color has landed on the retina. So, that is how color gets coded. So, detection of contrast and images is done lower level, that is basically done in the retina. Then primitive surfaces go all the way back to visual cortex. So, visual cortex, retina, then there is a lot of intermediate stuff which is happening at the LGB, which is position locking and coding and mixing of data from both the sides of the field. Then like in image processing. So, conventional image processing requires pre-processing of data, that is the engineering side of the study. So, pre-processing is basically already done, pre-processing is done in the retina, contrast enhancement, the color fine tuning, on off, all those are pre-processing kind of stuff. So, pre-processing is done in the retina, the image gets on to the LGB where spatial data is integrated, because field level data, this data is mixed specifically, reaches specifically the LGB, from there it is transferred onto the visual cortex. Visual cortex lines, things are drawn, and then you have an object recognition which says that there is a particular object. It can be linked with previous objects, say we have a vast storage of these things. So, maybe various kinds of objects are you map it onto your prior history. So, it is an exotic horse, it is not exotic horse, it is a breed which you recognize, breed you do not recognize, it is a horse generated by artificial intelligence, it is not an artificial intelligence generated horse, all that stuff. Then there may be categorical linking. So, categorical linking is whether it is a play tool, toy, different kind of toy. Then you can have associative linking. So, association of horse with a rider, horse on, meaning in a game, horse as such in a what is that is a play whatever arcade game. So, you make up associative linking, categorical linking, these are higher level stuff. So, starting from image we are going into higher level stuff. Starting from object representation. So, these representations are saved say partly in working memory, then integrated with signals from other modalities, say sound of a horse or smell of a toy, those things are connected, and then there is emotional valence. So, you may have an emotional history which you attribute or label this particular which you can give label, sorry, so an emotional label also gets attached. So, the next time or sometime in the future when you are recollecting the horse which you saw now or in this picture, you would also have the emotional baggage which you had labeled along with that object at that point of time, and that is how the it's seamless. The memories being associated and connected and shown over here are all seamless, and that's how vision is processed, starting from individual photons, generation of potentials, local pre-processing. I think pre-processing is too simple to be told to the activity. It is contrast enhancement, line recognition, object recognition, all those stuff happens over there, but it is parsed on up. You have spatial coding where from the object is obtained, and then it goes to the higher level cells where motion classification happens, image segmentation happens, multi-object classification happens, and along with that you add the emotional stuff. You take in stuff from memory and connect it to it either for future storage or working memory. Horse in the present environment is something to be handled, hostile, not hostile, friendly, unfriendly, able to ride, not able to ride, all those classifications are made at that point of time. So, that is the idea of this graph. So, I think we can I close the topic on vision at this place. We will continue with the next class on something else.